Hello, everyone, and welcome to Chapter 17. And regardless of what the notes say here, this is not the functional anatomy of prokaryotic and eukaryotic cells. This is adaptive immunity. So this is what we're going to be covering. In the previous session, we went over innate immunity, or immunity that you are born with, and we talked about some of those physical barriers, and we talked about how the phagocytosis takes place, and kind of those things that just happen for you that you're just born with. Now we're going to talk about how your body deals specifically with specific infections as it refers to microbes. So these next couple of slides here are just some of the learning objectives. And I won't go over all of them. Actually, I'm not going to go over them at all. I'm just going to let you all read them, and they're also posted on Blackboard as well as some things that we will be covering. So a lot of what we're going to be covering in this chapter is going to be very familiar to you, simply because we talked about much of it in Chapter 16 with the innate immunity. So we've talked about the importance of phagocytosis. We have talked about what a cytokine is. We've talked about physical barriers and chemicals and neutrophils. We even talked about how we have these lymphocytes that differentiate into T cells and B cells. So some of the terminology, some of the language, some of the things that we're going to cover are going to be familiar to you. But what separates innate immunity from adaptive immunity is the specificity of adaptive immunity. We call it adaptive immunity because your body adapts to its environment to be able to deal with these different types of microorganisms over time. So it's not something that you're born with. So you get the chicken pox one time, you're not very likely to get it again. And that sort of thing is the role or the responsibility of your adaptive immunity. So we basically are talking about one type of cell that differentiates into two different classes of cells called T cells and B cells. B cells originate or differentiate in the bone marrow, and the T cells differentiate in the thymus glands. The B cells are responsible for the production of antibodies, and those are made from plasma cells, and the T cells are responsible for what we call cellular-mediated immunity. So we have cytotoxic T cells, which we talked about a little bit in the last chapter. We have helper T cells, and we also have T cell regulatory T cells. So there are different camps of T cells that will do different jobs in order to maintain this immune response. And for both the T cells and the B cells, they also have memory cells. So humoral immunity, or what we refer to as the role of B cells, because they're made in the humors of the body and the bone marrow, the job of these B cells is that they are going to make antibodies. So we can call it either antibody-mediated immunity, or we can call it humoral immunity. So you kind of hear your book, we'll read your book and hear me toggle back and forth between antibody-mediated immunity, B cells, and humoral immunity, B cells. Cellular-mediated immunity is due to the T cells, and the T cells originate in the thymus gland. So an easy way to remember who originates where: B for bone cells, bone marrow, and T for T cells originate in the thymus gland. So once again, as I said before, some of this information is going to be pretty, you've heard it before we talked about it. So an antigen, remember, is a substance that causes the body to produce antibodies against it. All bacteria or viruses or any invading pathogens typically have cell surface markers that are different from your own cell surface markers, and we call them an antigen. Those antigens can be picked up by the fact that, hey, this isn't supposed to be here, not only by your innate immunity, but more specifically by your specific immunity, those T cells and those B cells. So your body can make specific antibodies, job of the B cells, or synthesize T cells to interact with this antigen. So antibodies are going to interact with what we call epitopes or antigenic determinants, or special segments of the antigen on the bacteria or the virus, or let's just say pathogen. A hapten molecule is an antigen combined with carrier molecules. And hapten molecules are just, they're part antigen and their part carrier molecule. And sometimes the antigen itself doesn't really trigger an immune response until this part antigen is happened, until it actually binds with a carrier molecule. Once it binds with a carrier molecule, which normally doesn't affect you, which could be some sort of protein that your body normally makes, if it doesn't, normally that carrier molecule doesn't affect you adversely until it binds with this partial antigen. When it does, then it elicits immune response. Good example of that would be penicillin allergies. So the penicillin that you ingest or take or are exposed to will react with some individuals' carrier molecules, a protein that's normally in their body, and causes them to elicit an immune response. So the first thing we'll talk about are antibodies. Antibodies, remember, are made by B cells. Another name for these globular proteins is called immunoglobulins. The number of antigen-binding sites determines the valence. So there are 5 different classes of antibodies: IgG, IgE, IgD, IgM, and IgA. But within those 5 different classes, there's a lot of variability. And even all the antigens in the IgG category are going to have slightly different number of valence, so that they'll be able to defend the body against various different types of microorganisms. So here's a picture of a typical antibody. We have our constant region here in the back for both the long chain, or the heavy chain, and the short chain, or the light chain in blue, heavy chain in green. And then at the tips of it, you have the variable regions. And these variable regions are where the antibody, this guy that we're looking at, will attach to the antigen on the surface of the microbe. So this region here is called a variable region because it changes. This constant region is going to stay the same for each antibody within that class. So everything in IgG is going to have the same constant region, everything in IgM has the same constant region as the others, and so forth. And we've talked about antibodies binding to antigens as a method of activating complement in Chapter 16. So this information isn't all 100% completely new, because we've talked about antigen-antibody complexes. So to activate these B cells, we have different kinds of markers that are found in your own cells. They're called major histocompatibility complex markers. They're expressed in all mammalian cells, and you have 2 classes of them, class 1 and class 2. T-dependent antigens are antigens presented with self-MHC markers, and really that's what this major histocompatibility complex marker is. Just in the same way that bacteria and viruses, those pathogens, they have their cell surface markers that says, hey, I'm Joe E. coli, the bacteria. Your own cells have special markers that say, hey, I'm a Kelly cell. So immune system, don't attack me. I'm supposed to be here. So those are those MHC markers. So antigens presented with self-MHC markers to helper T cells. And so if that marker is slightly changed, depending on whether it's class 1 or class 2, it could send a couple of different messages to the immune system. It could say that this is a sick cell, and it needs to be it's been infected by a virus or a bacteria, and it needs to be destroyed by the immune system, or it can act as an antigen-presenting cell and say, hey, this is the antigen that needs to be killed. And we'll talk about the differences between the 2 of those in just a few slides here. But what I really want you to take home right now is that these cell surface markers that are on your cells are called MHC markers. And those MHC markers help to tell the immune system if something is right or something's not right. And it does this through these chemical messengers called cytokines, and it activates B cells or it can activate T cells. So here we have an activation of B cells. So we have the antigenic fragments, so this would be from a bacteria or from a virus or whatever it is that's infected your body, and it binds to this antibody here. And when it binds to an antibody, it can become ingested by the cell, and then it's going to merge with this MHC marker. And this particular marker is a class 2 MHC marker. So for class 2 MHC markers, they're only found in very specific types of cells. They're found in phagocytotic cells. They're found in some classes of B cells. And so when I say some classes of lymphocytes, that's pretty much the same thing. So when the antigen, the thing that came from the pathogen, is digested by this cell, it's now acting as an antigen-presenting cell. And we call it an antigen-presenting cell because this MHC marker, or major histocompatibility marker, which is not found on everything, but only found on those cells such as phagocytotic cells and some classes of lymphocytes, mainly B cells, presents that to the immune system. So basically what this cell is saying is that, hey, this is what's making us sick. Is there any cell that is going to be able to deal with this infection, that can deal, that is specifically chemically developed to deal with this infection? So it's presenting this antigen to the rest of the immune cells. So the helper T cell can come along and recognize that perhaps that it is this cell that's presenting an antigen that can deal with it, and it will do what's called co-stimulation. And if it says, yep, you're right, you have the memory, you've seen this before, you're chemically developed and made to deal with this, it will activate this cell, this B cell, and tell it to make copies of itself to differentiate into memory cells and plasma cells, which will then make the antibodies. So this whole process of clonal selection and differentiation, we just looked at the kind of tail end of it all, but what determines which of these classes of B cells are going to be cloned or made lots of copies of, it just depends on which of those classes of B cells has the antibodies that fit that particular antigen. So remember, there's a lot of specificity in innate immunity. So which specific B cell is developed for this particular antigen. And it looks like in this case, for the diamond-shaped antigen, it's this antibody or this B cell number 3. So when it is triggered by the helper T cell through chemical messengers called cytokines, then it will tell this cell, yes, you are able to deal with this infection. It's kind of co-signing off on it making extra copies of itself, and we want to have this co-stimulation take place because we don't want to make a bunch of cells and use all this energy if they're not the right ones to fight this specific infection. So that's why we have this selection of which cells are going to get cloned. So once we have that cytokine, that chemical messenger, say yes, go ahead and make copies of your cells. They differentiate into 2 camps, memory camps that are going to be long-lived cells that can be used at a later date if we see this infection again. And then you have these active cells that become plasma cells, and the plasma cells of B cells are the ones that actively make the antibodies that are able to agglutinate or attach to that antigen or to deal with this infection. So B cells differentiate, as we said before, into 2 camps, antibody-producing plasma cells and memory cells. And just as we had clonal selection and differentiation, we're going to have clonal deletion that will eliminate harmful B cells. So B cells that we don't really need or that may act against the body, or if we've made too many of these B cells, they can elicit an autoimmune response where the body starts to attack itself. The immune system will attack different parts of the body. You want to get rid of those. So the results of antigen-antibody binding, some of which we've talked about before. This opsonization, when we were in complement in chapter 16, we said it helps to make this bacteria sticky, specifically if it has a capsule. Bacteria that have a capsule are slippery and they're kind of hard to get a hold of. So with the antibodies attaching to those antigens on the surface of the bacteria, it helps to enhance phagocytosis by making it stickier so this phagocytotic cell can get to it. Agglutination helps to reduce the number of infectious units so that they can be phagocytized or dealt with easier. And we have neutralization, which can keep the bacteria or the virus or the toxin from getting into the cell and causing damage. The activation of complement, this is also something we've seen from Chapter 16, that if we have an antigen binding to the antibodies, then it's the classical method or mode of activating complement. It can also, which will lead to lysis, more optimization, or inflammation. And then we have antibody-dependent cell-mediated toxicity, where the antibodies will interact with this target cell and cause destruction by the macrophages or eosinophils from the outside in because those antibodies are attached to it. So these are all different things that happen as a result of these antibodies now circulating in the bloodstream. And remember, we don't just pick any antibody willy-nilly. We have to make sure that the antibody that's chosen to be cloned specifically fits the infection or the antigen that the body is encountering. So now on to cellular-mediated immunity, and that would be the role of T cells. And we have different classes of T cells. One T cell we've already talked about before, when we said that this T cell helps to activate those B cells and make sure that it gets through co-stimulation, it helps to make sure it gets activated properly. So it activates cells related to cell-mediated immunity, macrophages, T cells, and natural killer cells, helper T cells too, stimulates production of eosinophils, IgM and IgE, so that's kind of what we looked at with the B cell activation and clonal selection and differentiation. Cytotoxic T cells, their job is to destroy target cells on contact. Regulatory T cells are going to regulate the immune response so that after the initial immune response, we don't want to have all of these cytotoxic T cells roaming around because it could elicit an autoimmune disease or an autoimmune response where the body starts to attack itself. So it helps to kind of regulate those numbers. Active macrophages and natural killer cells, they enhance phagocytotic activity. They also will attack your own cells if they start to go rogue, that we call cancer cells. And then natural killer cells, they kind of fit into both classes of specific immunity and non-specific immunity because they can attack and destroy target cells if they don't have the right MHC marker on them, and it can also participate in antibody-dependent cellular-mediated toxicity, which we saw in the previous slide, with dealing and how your body can deal with parasitic worms. So T cells respond to antigen by what we call whole cell receptors or T cell receptors. T cells require antigen-presenting cells. The pathogens that are entering it through your gastrointestinal tract or respiratory tract, they're going to pass over a second class of cells because you're introducing these things from the outside of your body to the inside of your body, so they're going to be loaded with bacteria. And you have microfold cells and Peyer's patches that also contain antigen-presenting cells. Just as we saw in our very first discussion when we talked about the B cells, that those B cells can sometimes act as antigen-presenting cells. The key feature on whether or not a cell is an antigen-presenting cell is whether it has that major histocompatibility complex, that MHC marker 2. So if it has an MHC2 marker, then we consider it a type of antigen-presenting cells. But there are macrophages, B cells, and like I said, lymphatic cells that can act as antigen-presenting cells. So CD4 lymphocytes, or helper T cells, their job is to, start starring some things because it's kind of important, is to recognize antigens on MHC2 markers. Remember, we said the importance of that. And if the MHC2 marker has an antigen, we call that an antigen-presenting cell. That's saying this is what we need to fight. Which of you T cells can deal with this particular antigen? Toll-like receptors or toll-like receptors are costimulatory signals that will be on the antigen-presenting cell and the helper T cell. So then the helper T cell will produce chemical messengers that will tell the cell that it can fight the infection to divide or differentiate into different classes of the cell. And also we have our campus memory cells. So other types of T cells, remember these different classes of T cells. They're great for short answer questions and fill in the blank. T helper cells, 1, produce interferon gamma, which activates cells related to cellular-mediated immunity. antibodies and macrophages. Helper cells 2 activate eosinophils and B cells to produce IgE, and helper cells 2 we talked about with the B cell activation. T helper cell 17 stimulates the innate immune system and to get those macrophages and enhances phagocytotic activity and all the things that need to happen with complement. And then we have TF helper T cells that will stimulate B cells to produce plasma cells and are involved in what we call class switching, which is basically switching different classes of antibodies to be activated. So here we have the activation of the CD helper T cells. We have the antigen that's being phagocytized by the dendritic cell. The antigenic fragments are going to be displayed on the class 2 MHC marker. This toll-like receptor recognizes that, and it's going to release chemical messengers to activate another cell, a cytotoxic T cell, to be activated so that it can kill the specific infection. So CD8 cells, also known as cytotoxic T cells, they're going to target cells that are carrying endogenous antigens, which means that it's an antigen that has made this cell sick. So what these cytotoxic T cells can recognize is that they can recognize when an antigen is on a different kind of marker. So instead of it being an antigen on MHC2 marker that says, hey, this is what we need to kill, antigens that are presented on an MHC1 marker say, kill me, I'm sick, I have an infection. So when a cytotoxic lymphocyte recognizes that, then it will induce apoptosis in that target cell. So it will tell that cell, yes, you're right, you're sick, and it will have that cell undergo programmed cell death. Or the cytotoxic T cell can also release performins and granzymes, which will kill the cell from the outside in. So here, notice this marker looks a little bit different than what we saw for the MHC2 marker. If the antigen is on an MHC class 1 marker, the cytotoxic T cell receptors recognize it and say, yes, you are infected, and so then it will send chemicals or signals to this target cell to be killed. So this could not only be a cell that's infected by a pathogen, but this can also be a cell that is a cancer cell. So it's MHC class 1 marker, which just usually says, hey, I'm a Kelly cell, I'm okay, if it's naked by itself. But when it's attached to an antigen, or if it's slightly different from what it needs to be, the cytotoxic T cells will recognize that, and they will target that cell for destruction. Regulatory C cells have CD4 markers as well as CD25 markers on the surface, and remember, their job is to regulate the immune response, and they will suppress the number of T cells that are made against the body. Antigen-presenting cells, they will display the antigen on an MHC2 marker. Types of cells that act as antigen-presenting cells are B cells, which we talked about at the beginning of this session, dendritic cells, which we saw just a second ago with the activation of helper T cells, and then activated macrophages. They are the only cells that have a class 2 MHC marker. Every other cell in your body that has a nucleus has a class 1 marker. So if there's an antigen on those class 1 markers, then it tells the immune system that this, I am sick. If there's an antigen on a class 2 marker, and let's write that right here because that should say 2. 2. If it's on a class 2 marker, then it says, kill this. This is what's sick. This is what's making us sick. Natural killer cells destroy cells that don't express the right MHC1 marker. So if I give you my cornea and you don't take immunosuppressive drugs, cornea is the outer portion of my eye, or if I give you a kidney or whatever, I have different MHC1 proteins markers of my cells than you do, so your body will have an immune response to it, and that will be in part the job of natural killer cells. They can also kill virus-infected cells or tumor-infected cells simply because that MHC1 marker is not quite right, as well as attack parasites. Cytokines, we've been talking about them quite a bit this session. They're just chemical messengers, fancy way of saying chemical messengers. If you have too many of them, it can produce what we call a cytokine storm, and the body will be unable to differentiate what the message is, and your immune response will be compromised as a result. So different types of interleukins we have here, things you want to make sure that you recognize and that you take home from this chapter for your upcoming exam. Make sure you understand antigen presentation. Make sure you understand the differences between MHC2 and MHC1 markers. Make sure you understand how T cells are activated. Make sure you understand how B cells are activated, because those would be good short answer questions. So different types of cytokines, make sure you know those. Once again, great questions for multiple choice and fill in the blank. Interleukin 1 stimulates T cells. Interleukin 2, the proliferation of CD4 helper T cells and the proliferation of B cells. Interleukin 12 inhibits B cells and activates T cell immunity or cellular mediated immunity. Cytokines are chemical messengers. They're going to induce the migration of leukocytes. That's one of the things that they can do. These interferons, I'm more concerned with you remembering, alpha and beta respond to viral infections, and they will interfere with protein synthesis within the cell so that the virus can't make copies of itself. And then gamma interferons are going to stimulate macrophage activity. Immunological memory, your body kind of has that, lives by that old adage, fool me once, shame on you, fool me twice, shame on me, so that the next time that it sees this infection, it's going to elicit a more robust immune response, and it's going to happen quicker. Antibody titer is the amount of antibody in a serum, and it lets your body know whether, or lets your physicians know whether or not your body has seen this particular infection before. Your primary response is going to happen after your initial contact with the antigen. Your secondary response, or your memory, occurs the next time that you've seen it. And as I said before, typically your memory is so effective that you either don't get sick or feel the malaise of being sick, or you don't feel ill for very long. So when we look at this chart here, some things that I want you to pay attention to is that with your initial exposure, so we have our two different exposure times here, so that the first time that you see this infection, the response isn't very high, so the antibodies in your titer aren't super high. The second time that you see the infection, the antibody that you see first on the scene is IgG, and then IgM happens next. The very first time you see it, it's IgM and then IgG. These are produced by those B cells. So understanding that the first time that you see it, your titer of the antibody is not super high after your first infection, but the second time you see it, we see very quickly we have these IgG antibodies are activated in response to this infection, and you have a much higher level of the antibodies in your serum because you've got those memory cells that have just been laying in wait for the next time you see this infection. So there are different types of what we call adaptive immunity. We have naturally acquired and artificially acquired, and they both fit into the categories of active or passive. Naturally acquired active immunity means that you got sick. You actually got the chicken pox at a pox party. If you don't know what a pox party is, it's basically where your parents send you to another kid's house that has chicken pox, and there are a bunch of other kids there too because your mom wants you to get chicken pox now and get it out of the way, and the pox party. So you get the infection, and then you actively make your own antibodies as a result of that infection. Naturally acquired passive immunity means that you get these antibodies, but you didn't make them yourself. So you just receive them through the placenta if you're a fetus, or through the colostrum, or through the breast milk. So we can pass those antibodies on. So for active immunity, you're actively making your own antibodies. Passive immunity, you get someone else's antibodies. Naturally, you just got it in the wild. You just got it from being around the little children or whatever. Or you can have it naturally through the placenta, or if you're a fetus, or through the breast milk. Artificially acquired active immunity This means that you still, since it's active immunity, you're still going to make your own antibodies, but instead of just getting sick at a pox party or just getting sick somewhere, you were given a vaccination. So you were injected with the antigenic fragments or portions of the antigenic fragments, and that's what elicit an immune response, or how you actively made your own antibodies. With artificially acquired passive immunity still means you got someone else's antibodies, and this is an important slide to remember too. So make sure that you're familiar with this as well. You got someone else's antibodies, and you didn't get them naturally through the placenta or through the breast milk. You received an injection of that. And that is a kind of a classic treatment we have for rabies, is to give the antibodies that have been made by like a rabbit or a horse in their serum, and give those to the patient. All right, so that's the end of Chapter 17, and we will see you soon. Bye.